Good morning, friends. Today we'll be talking about very important aspects in paleontology, and that is called as the functional morphology. Each living individual, each living being has a particular function. And since the individual basically is a system, the body system, and this system has many components. Like in our body, we have hands, legs, heart, kidney, liver, many organs, and each organ is having a function. That means each morphological feature in a particular individual or an organism is imparted a very specific function, and that a particular organism performs different functions. So, for different functions, it has different organs. Sometimes an organ performs more than one function. So, the entire function of a particular individual is a sum total of the function of its specific organs. So, we have function at the level of an individual and the function at the level of its morphological part. My function as a total is different than the function of my heart. My function is totally different than the function of my digestive parts. So, we have functions occurring at the level of an individual, and the individual's function is supported by the function of its morphological parts. In paleontology, you may be thinking what use this discussion has in paleontology. Actually, I will say that in paleontology, we get organisms preserved as fossils. And to be very precise, we get mostly hard parts of that organism which is preserved as fossils. Soft parts are normally not preserved. That means you can, estimate or you can judge the function of a particular morphological feature in spite of judging the function of the total organism. That means organisms function in totality cannot be judged in fossil forms. Why? Because the entire organism is not preserved. Only a part of that organism is preserved, and mostly that is hard part. And even that will be called, it is partial, I understand, but then this will be also called as a success if we are able to identify the morphological parts and assign a function to it. In a living being, in a living form, when the organism is living, this particular task is very simple and easy. Why? Because you can see the individual functioning in the present time, and the morphological feature that the particular individual exhibits also has certain functions, and that functions can be observed, can be studied. But in fossil forms, where the preservation is very partial, entire organism is not preserved, only some part of that organism is preserved. So, seeing that fossil form, assessing the function is little difficult, and this difficulty is added by another difficulty, which is that a particular uh, organ or a particular morphological feature performs more than one function, and one function is performed by more than one morphological features also. In that case, there can be one approach that is usually followed, and that approach is to compare the fossil form with the modern counterparts. I mean to say that the particular fossil forms has certain morphological features, and what morphological features are present in the present form, they are compared. The first comparison is on the morphological features. Then the this morphological feature which is present in the living form, it performs what kind of function is the second level of comparison that any paleontologist would imply. I mean to say that the function which the living organism performs, or the part or the morphological feature of that living organism performs, is compared with the similar structure in the fossils. So, this is a kind of a comparison, two-step comparison, and it is by this two-step comparison that we can understand the morphological function of a particular feature in the fossil forms. Now, this comparison has also certain grounds. For example, I select a morphological feature in a living form, and I select the same morphological feature in the fossil form. That means the prerequisite for this comparison is that I should have a fossil form for which the living counterpart is available. Then the morphological feature in the living form performs some function. So, the same function would be assigned to the same morphological feature in the fossil form. But this part is not as straightforward as it looks in the first sight. That means when the organism has the morphological part in the present time and it performs certain functions, then the morphological feature in the fossil part is believed to perform the same function based on the fact that the present is key to the past. But to have that understanding, we must assume that these structures have the same genetics or the same origin. That means the two structures have in the organisms having the fossil and the modern counterpart, and these two structures have the same origin and they perform the same function. So, they should perform the same function, they should have the same origin, and the organism which contains that morphological part in the living state and in the fossil state must be related. So, if all these things are in line, then we can say that we are performing the functional morphology. So, what I am discussing through the discussion today, through the facts that we are taking into account, is basically the functional morphology. And continuing our discussion in that line, we have so far discussed that the particular structure in the fossil state, the particular structure in the living state should perform the same function. That means this is how you can understand the function of a morphological feature in the fossil state. But if they have the same origin, performs the same function, then such structures are called as homologous structure, and this process is called as homology. That means for the two structures to be homologous, they must have the same origin and they must perform the same function. But then comes another part. Sometimes these structures that have the same origin, they doesn't perform the same function. For example, in the fishes, if you see, so fishes have the limbs, but these limbs have the same origin like our hands and legs, but they don't perform the same function. There the limbs are used for swimming, and in us the limbs are some two of the limbs are used for walking, while the two hand limbs, they are used for other purposes. In a similar way, that means these are the structures having the same origin, but they don't perform the same function. So, this will also come under homology. But the true homology is when the structures have the same origin and perform the same function. On the other hand, sometimes these structures may perform the same function but have the different origin. For example, the wings in a bat and the wings in a bird. The wings of the bat are used for flying and wings of the bird is used for flying. These are the two structures which perform the same function, but they do not have the same origin. The bird is an ave, have a different phylum, and the bat is a mammal. So, the wings of the bat, it's like the limbs of our hands. So, the origin of the wings of a bat is equivalent to the limbs of our hand, because both of us are mammals. But it resembles with that of the wings of a bird, to which it is not genetically related. That means the wings of the bird and the wings of the bat, performing same function but has different origin. Such structures are called as homoplasic structures, and this function is called as homoplasy. So, I have described two functions, homology and homoplasy. In homology, you have the same function and same origin, and in homoplasy, we have the same function but not the same origin. That means here in homoplasy, the functions are only superficial. Why superficial? Because they do not have the same origin. But the true homology is when they perform the same function and has the same origin. I hope you would have understood these 2 points. Moving ahead, we can find that the homology or homoplasy, it is basically our way of comparison. This point is to be understood that it does not exist naturally. The natural existence is different, and this is only our personal way of comparison of these naturally existing traits. The part that is to be performed here, actually the basis for such homology or homoplasic comparisons is that a particular fossil form that we see in paleontology should have the modern counterpart. That means if the fossil has no modern counterparts, the homologic or homoplasic approaches are very difficult to be applied. But before I dwell deeper into that, let me tell you a formal procedure of applying homology or homoplasy. The formal procedure is that in a particular fossil form, a particular morphological feature is selected. That means select any morphological feature in the fossils you have obtained, and then the function, the hypothetical function of that particular morphological feature is assumed. Then the living counterpart of that particular organism is also selected, and the same morphology is compared with the function it performs now. We also see the function of that particular morphological feature in that organism in the light of its ecology. That means its habit and its habitat. That means whether the function that which particular morphological feature performs is suitable to its ecological habit or habitat or not. This is the second step of our approach. And if that step is also clear, that means I have selected the morphological feature in the fossil, I have selected the same morphological feature in the same organism in the living counterpart, I have hypothesized or I have assumed a particular function of that morphological feature, and then I have also compared and correlated that particular function of the morphology in its ecosystem or in the light of its habit and habitat. Then that particular function to the particular structure is fixed for that particular fossil form, either by homology, that means the same function and same origin, or by homoplasic approaches, that means the function is superficially similar but they have the different origin. And this finishes our first part of our discussion, where the fossil forms has the modern counterpart. But the second part of our discussion is related, or it is about the forms that do not have any modern counterparts. How to understand the function of a particular morphological feature or the structure in the preserved part of an organism when you do not have any modern counterparts. In such cases, the paradigm approach is used. Paradigm approach was proposed by Redwick in 1964. What is paradigm approach? This is our next part of the discussion. Paradigm approach is basically the approach in which we assume or we assign more than one function to the particular morphological feature in consideration. And each particular function to that particular morphological feature is called as a paradigm. That means we have more than one paradigm for a particular morphological feature. This is reviewed in the light of its ecological niche, its ecological habit or habitat, and then for that particular structure different functions or different paradigms are compared, and the paradigm which suits much with the ecology or the the habit or habitat of that particular organism is selected, and that paradigm is used to assign the function of that particular morphological feature in that fossil. The success of this approach is that even if the modern counterparts are not present, this approach is successful in assigning the function to a morphological feature. But this paradigm approach has to be applied with due care and caution. The caution is that this particular paradigm or the approach which we have used to assign the function of a morphological feature of a preserved part of a fossil should be tallied or should be correlated with the physiology and genetics of that particular fossil or the group which to which that fossil belongs. Otherwise, all our efforts would be insignificant, to be very frank. After discussing homology, homoplasy and paradigm approach, let us discuss few parts which is very essential for any paleontological discussion, is that the line of evolution of a particular life form, either as the modern forms or as the fossil forms, is a kind of a compromise. And you may be thinking why it is a compromise. Let me tell you. Consider a particular mollusk, any seashell. That means the soft part of the body enclosed within two shells. In Hindi, you can call it as ghonga and seep. That means, what is that organism? Organism is a soft part, and that soft part is enclosed within a pair of shells which is enclosing it. Nature has given us the body skeleton, but that skeleton exists inside our body, that is called as endoskeleton. But for them, those shells, the skeleton exists outside their body, that is called as exoskeleton. We have our nervous systems well developed, we can think, so we can protect ourselves. So we do not need skeleton for our protection, we can use our brain, our nervous system for our protection. But that is not well developed in those organisms, because of which the nature has given them exoskeleton. So the skeleton themselves protect them. But then where is the compromise which I was discussing? The compromise is that this particular external shell in those mollusks, actually it does two functions. First function is that it provides the framework for the soft musculature, soft parts, and then it provides the safety of that particular organism with the predators. So it is serving two functions. It is providing you two functions. First, the support of the musculature, and then safety. So, support and safety are two functions which the external shell does. So, that means a particular morphological feature can perform more than one function. But then again, where is compromise? The compromise is when the shell is thicker, when you have thick shells, it will be very difficult for the predator to puncture that. That means it will be providing much more safety to that organism, to that shell. But then if you thicken the shell, it will be difficult for that shell to move and run away from predators. And what organism can run away? Those who have thin shells. But then thin shells help in escaping from the predators, but thin shells are very prone to be punctured. So, there is a safety and unsafety if the shells are thicker. There is a safety and unsafety if the shells are thin. So, the particular shell thickness that is to be given to a particular organism, it's a kind of a compromise. And each particular mollusk or the lamellibranchs, they have different thicknesses of the shells, and that thickness is a kind of a compromise between the ecological constraints. The constraint is that the thicker shell will provide safety, but it will make the organism difficult to escape because of that heavy weight of that shell. And thin shell will provide you the easiest task and buoyancy to run away from the predator. But then if the shell is thinner, it will be vulnerable for the strength of the shell for the protection. So, this is one sort of the compromise. The second sort of the compromise, by the other example in the same group I will be discussing, is that for the shells to open, and this is an example I will be taking from the lamellibranchs, if the shells are opening, the umbones means the highest dorsal part of the shell is called as umbo, to be reminding you, and the umbones have to be little far apart, only then the shells can open. And to make the umbones far apart, the ligament has to be stretched. So, if you overstretch the ligament, it will be broken. That means the ligament, if you are stretching the shell, making the umbones apart and stretching the ligament will be broken. And if you relax the ligament, the umbones will become much closer, and that will obstruct the closing of the opening of the shell. Again, here the structures are conflicting, but the shells has to be open. So, the particular mechanism an organism at the level of an individual or a group would acquire will be again a kind of a compromise. Now, this compromise is resolved as seen in the paleontological record through adaptations and several lines of adaptations. If you see all the Paleozoic lamellibranchs, they are all equivalve mostly. That means if the valve size is unequal, then this particular conflict can be resolved. So, you can see many, and the dominance of the Mesozoic lamellibranchs, they are inequivalve. So, this is one line of the adaptation, but this line of adaptation is confined to a particular lineage. Then comes another line, that means the ligaments are replaced. The replacement of the ligaments, if the ligaments are broken, they are replaced. So, this is another kind of adaptation that has appeared in a different lineage. Then the umbones became far apart. This is third line of adaptation, and it appeared in a different lineage. So, on the whole, the adaptations were followed, but these adaptations are lineage specific, and only through the paleontological record such changes in the geological histories can be understood in terms of evolution. And regarding the other features like hinge and the muscle scars and other morphological features, these morphological features are basically related to the ecological balance or the ecological constraints imposed on a particular individual, but they do not have much to say as far as the adaptations are concerned. So, I have discussed today the idea of functional morphology in relation to the homology, homoplasy, the formal method of the application of the homology and homoplasy, the paradigm approach when the modern counterparts are not available, and the evolution as a kind of a compromise with the help of two examples. I hope that clears the concept of functional morphology in paleontology in your mind. We'll be having the discussion for interesting discussions for the more paleontological groups in the lectures to come. Till then, thank you so much.